Good afternoon. Thank you for coming to hear about Mitotech. My name is Natalia Perekhvatova. I'm the CEO of the company. We are a privately held small molecule company. At the heart of our technology is SKQ1 molecule, which is a mitochondria-targeted antioxidant designed to scavenge excessive ROS reactive oxygen species inside mitochondria. It was designed as part of an anti-aging research and has proved to be a safe and potent drug candidate for many age-related and mitochondrial diseases. So to date, we developed two formulations, eye drops, where we are at clinical stage, and oral formulation, where we target bigger markets, but we are at an earlier preclinical stage. So dry eye disease is our most advanced indication. We recently launched pivotal phase 3 study in the US for ALHON, rare mitochondrial disease. We received positive clinical data from a study done in Russia, and we are ready to initiate phase 2 in the US. For dry AMD, we have very positive animal data and are also phase 2 ready. For oral administration, we completed IND toxicology program, no negative findings there, and we are phase 1 ready for indications for NASH and multiple sclerosis. So the company started its drug development program in 2010. We went through preclinical studies and completed phase 2A in partnership with Aura in Boston, Massachusetts. In 2018, we secured co-development financing deal of approximately $40 million to take us through phase 2B and phase 3 for dry eye indication. So worth noting that we are also on the market in Russia for a few years and recently started clinical development program in China for dry eye. So this is our lead molecule. It is a powerful mitochondrially targeted antioxidant. It has two parts. One is a positively charged ion designed to get inside mitochondria, and the other part is an antioxidant itself, which scavenges for ROS once the molecule gets inside mitochondria. It has a novel, unique mitochondrial mechanism of action. In addition to scavenging ROS, it was shown to protect cardiolipin in mitochondrial membrane against oxidative stress, and it counters mitochondrial dysfunction, cell apoptosis, and quite unexpectedly, it also had, we saw that it has an anti-inflammation effect. So to demonstrate this mechanism of action and also to show the impact on the lifespan of living organisms, we conducted a series of animal experiments. One of them was conducted at Stockholm University in Sweden. There were over 50 mutated mice, and it was shown that SKQ1 protected against cardiolipin depletion in multiple tissues. We can see that it was statistically significant in skeletal muscle and liver. Also, mice were shown to increase their lifespan significantly by 15 to 20%, and we also did see multiple health benefits on mice in that experiment. So when looking for an indication, we decided to tackle dry eye disease. Similar as SKQ1 mechanism of action, it is a multifactorial disease that is characterized by different signs and symptoms. Inflammation is one of them, but it's also gland tissue degeneration, tear deficiency, corneal damage, and other signs and symptoms. So it's also a growing market. It is expected to grow to $7-8 billion US dollars in 2025. It has only two active RX ingredients on the market today. It's cyclosporine and Xiidra. They both tackle only one aspect of the disease, which is inflammation, and do have some tolerability issues. So there is clearly in need for a better drug on the market, and SKQ1 is well positioned to be just that drug. In addition of inflammation, its mechanism of action allows to target other aspects of the disease. And it has an excellent tolerability profile, unlike the other players in the market. So a few words about our phase 2b study that we recently completed in the US. It was multi-center, double-masked, randomized, placebo-controlled study. It was run in 7 centers across the US. There were 3 arms, higher concentration and lower concentration, that were run against vehicle, which is in our case artificial tear, about 150 patients per arm. So the results, it was a 2 months treatment administered twice daily. We had very positive results of that study. So especially for higher dose, SKQ1 reached statistically significant effect on multiple signs and symptoms, which is very rare for dry eye disease. Conjunctival fluorescent staining and ocular discomfort were the leading endpoints in terms of separation from placebo. We didn't only look at clinical study data, we also compared the data with historical data available on a market leader. Xiidra is a Novartis drug. So what we were pleased to see that SKQ1 showed twice the separation from placebo in a third time compared to Xiidra. So just at 4 weeks compared to about 12 weeks. So with these results, we initiated pivotal phase 3 study in the US. We initiated in December last year. So it's the same duration of treatment, same administration, now just 2 arms, higher concentration and vehicle, 300 subjects per arm. And we expect to get top-line data in Q3 2020. So in addition to our ophthalmic indications, we've also been working on showing safety and efficacy for an oral administration. So we completed IND toxicology program, no negative findings there. And this is run multiple animals models to show its efficacy. This is an example for EAE mouse model. We ran it against SKQ1, against vehicle and dexamethasone, and SKQ1 showed performed statistically indifferent, statistically the same as dexamethasone. Both drugs suppressed clinical symptoms completely, but SKQ1 has done so without the negative side effect of a steroid. Another example of SKQ efficiency is a STEM mouse model, a golden standard for NASH model, for NASH disease. So SKQ1 demonstrated dose-dependent effect on both clinically relevant disease scores and biomarkers of oxidative stress in that one. So once we get the results of phase 3 that we're obviously looking forward, we want to out-license rights for dry eye disease and use these proceeds to progress with clinical programs for other ophthalmic indications, Alhon and dry AMD, out-licensing them as we progress along the clinical programs. For systemic formulations, we are looking now to attract funds to take the drug through phase 2A for NASH and multiple sclerosis, and looking to an IPO to finance late-stage clinical development and commercialization. So once we do build a strong pipeline, we would like to go back to our anti-aging origins and develop low-dose formulation, but this is part of our longer-term strategy.